All right, good morning, everybody. Thank you to all of you that are obviously spending some brain cells trying to figure out these questions. It's clear that Mendel's second law is not something that's intuitive, straightforward, easy, sometimes even understandable. So that's what, I'm glad I asked these questions on this quiz. I've learned some interesting things, and now we're going to move forward with solving some problems together. So you might notice off the bat that I've got many fewer slides in this presentation than I normally do. And that's because today is all about just doing extra work on understanding and using and employing the second law, Mendel's second law, which all of these questions had to do with. Before we get into that, though, does anybody have any questions about non-second law related material? Super. So results, see, question number one, I guess that's kind of irrelevant. Question number two, and by the way, if you just walked in, please do the Socrative quiz. So the Socrative quiz today will be graded. So start it now if you want to walk through it with us. It's just for attendance, you know that. And also a good opportunity for you to get feedback. So dihybrid genotype, what are we looking for? About 2/3 of you got this one right. So what are we looking for when we look for dihybrid genotype? So two di, we're looking for two genes, and then hybrid indicates heterozygosity. So how do we know how many genes we're dealing with in a genotype? What's the character up there that tells us whether or not we're dealing with alleles or genes? Pardon?
Semicolon.
Okay, so the semicolon is what tells us. This is what separates genes. So anytime we see a semicolon, we know we've got, in this case, one set of genes, or one gene, a second gene. So there's the A gene, there's the B gene. What does the slash separate? Semicolon tells us we're dealing with two different genes, the A gene and the B gene. What are the slashes for? Alleles, absolutely. So we have two alleles at A, heterozygote, but in this case, B is homozygous, so not a dihybrid. If this was going to be a dihybrid, we'd have to make one of those big B's a little b. Now we have a dihybrid. Two genes, each of those genes has two different alleles, a big A and a little a, and a big B and a little b. And by the way, that is answer E. So E is a dihybrid. Is there any other dihybrid up here? C is a dihybrid. So just because I'm using plus minus notation doesn't mean that that's not a heterozygote. That's still a heterozygote. So C counts as a dihybrid. Two genes, there's one semicolon, so it's separating two genes that we're talking about, and each of those genes is heterozygous. Why isn't B an answer, a correct answer? There's no semicolon in the middle, instead there's a slash. So this is the genotype of a tetraploid organism. So if an individual has 4 copies of every chromosome, that would be a genotype, for example, which is kind of weird because why would we have two different sets of letters representing one gene? But that's a genotype. There's no semicolon. So you would have to assume that we're talking about 4 different letters that were all referring to the same gene, even though they're different letters, A's and B's. So that was the distractor, not a dihybrid. Questions about this? 2 genes, 2 alleles, that's a dihybrid. Okay, question 3. We won't go over. Everybody, almost everybody, chose either B or D or both. It's both in this case, B and D. So maybe you didn't look through all of the options or you didn't think about choosing all that apply. But you've got 2 genes. So you can either combine the big B with what's certainly to be a little d at the D gene, or you can combine the little b with one of the little d alleles. So whenever you have a heterozygote, you make twice as many types of alleles, or twice as many haplotypes of gametes as you would from a homozygote. And that's what we use to answer question 4, which has the hugest variety of answers. 2, 4, 5, 7, 9, 10, 6. So we're all over the place on the next question, and I'm not surprised. This comprises the bulk of what we're going to be talking about today. This is Mendel's second law in action. If you have an individual that's got, how many different genes are we working on here? Question 4. 5. So we've got the A gene, the B gene, the D gene, the F gene, and the G gene, all separated by semicolons. So how many different haplotypes, how many different gamete genotypes are going to be made with those alleles? 5 different genes, all independently assorting. Mendel's second law. What could you guarantee me right now about every single allele, or every single, haplotype that's produced by meiosis from this cell? What can you guarantee me about what alleles it will have? There is a big, long blue cell. It's got that genotype. It goes through meiosis. We're going to gamble money. What alleles will this cell, after going through the 2 divisions of meiosis, certainly have? Bless you.
The homozygous?
So if for a homozygous genotype, like big A over big A, at the A gene, what can you guarantee me that the gamete is going to be carrying? Big A or little a? It's going to have big A. There's only one choice. So 100% of the gametes are going to have a big A. Everybody okay with that? So homozygous, the diploid cell's homozygous, so it can only give gametes little big A's. What happens when we get to the B gene? Now we've got a heterozygote. So each of those 100% of cells carrying big A is either going to have what at the B gene? Half of those A's are going to have big B's. Half are going to have little b's. Because we've seen when you do meiosis with a heterozygote, half of the gametes get the big B, half of the gametes get the little b. So at this point, we're up to 50%. 100% of the gametes have a big A, but only half of them have a big B, the other half have a little b. So now we're making 2 types of gametes, big A with big B and big A with little b. What happens when we go to gene C? Oh, sorry. You can't tell my uppercase and lowercase c's, which is why I avoided using C. What about D? It's homozygous for D, so every one of the combinations of alleles we've drawn up there right now is going to be paired with, for sure, a D. Have to be. So all of those big A, big B carrying cells, the gametes, have to get a little d. There's no choice. This is independent assortment. For every different gene, whether or not you get one allele or the other is random, 50-50. What happens when we move on to F? It's homozygous. Every cell is going to have an F, capital F in this case. So to this point, we started out with all of our gametes we know are going to have a big A. Half of them have a big B, half a little b. All of them have little d's and big F's. What now for the last step? Half of the big A, big B, little d, big F either get big G or little g. The same is true down here. So we might wind up ultimately making 4 different types of gametes. That's one of the haplotypes, big A, big B, little d, big F, big G. There's also a type that has that same combination but with little g, and then we go down. So even though we're talking about 5 different genes, the fact that some of these genes are homozygous means they don't contribute to the diversity of haplotypes that get put into gametes. So we really only wind up making 4 different types of gametes. Every one of them has a big A, a little d, and a big F, because those were the homozygous genotypes that we started with. Half are big B, little b, and half of each of those are big F, or sorry, big G or little g. Am I going to ask you to set up a Punnett square? No. Bless you. I'm a little allergic to Punnett squares. I don't blame you. How large of a Punnett square would that be, though? Five genes. We do 1 gene. If it's a monohybrid, it's a 2 by 2 Punnett square. So what about this individual?
Square or rectangle?
Ooh, good question. Punnett square or Punnett rectangle? Well, in this case, how many different gamete types does each parent produce? Or one parent? We've got 4 different combinations up there. So there would be 4 rows and there would be 4 columns in this Punnett square. So it would be a 4 by 4 Punnett square. I won't draw it out here, but if you're interested, I can post a key to that problem later if you want. Second law. Question 5, it looked like most of you got the right answer. A, just briefly. That's because it was defined down here that the wild type allele is big R. And when I tell you that there's a plant with genotype little y over little y, and I don't tell you anything about what's happening at another gene, the gene that confers round or wrinkly peas, what was the assumption we're supposed to make? I told you the genotype of this plant here was little y over little y. So it makes yellow peas. What happened I told you about its genotype? That's what we're trying to infer. If I don't mention what's happening at the R locus, what do you assume? How do I know it's big R over big R? Or plus over plus? What's true about those R over R, big R over big R, or plus over plus? These are the wild type alleles. So we discussed that last class. Anytime you have a genotype and you don't know, I don't mention, you have to assume what's happening at a different gene. What's the genotype there? It's always going to be the wild type unless something else is specified. And in this case, I actually gave the information in the question. It would be a lot more complicated if I asked you to tell me which one was wild type. It's a separate question. So we move on into the one key point about Mendel's second law, optimizing cross designs. You have a solution, a goal in mind. You want to combine traits from individuals. We've been working with green fluorescent rolling worms. I wanted to remind you, now that we've gone through the exam or the quiz a little bit, why it's true that independent assortment works. What's the mechanism behind it? What's happening inside the cell that causes genes to independently assort? So this is from last class, if you have what's the genotype of that cell? Big A over little a, big B over little b. That's a what genotype? Heterozygous at two loci? Dihybrid, yeah. So it's a dihybrid cell. It goes through synthesis. We did this last class. So the big A duplicates, the little a duplicates, the big B duplicates, the little b duplicates. The reason I'm doing this again is because this is critical to the understanding of why the second law works. I didn't summarize it like this last class, plus as you know, because I admitted I failed to record this, and I don't know yet how many of you are taking advantage of those recordings. But just so it's recorded, here's the quick summary. How do you align all of these chromosomes at metaphase of meiosis 1, when the 4 copies of every homolog, the 4 A's and the 4 B's, line up across where the cell is about to undergo the first division? So you form a tetrad. All 4 of the A chromosomes come together in a group, like your 4 fingers lining up next to each other. So how do we draw the B's in? Review. Do the big B's necessarily go on the left side? It turns out no. So it could be by chance. I mean, the chromosomes are just floating around in the nucleus. The cell doesn't know how they're lining up. All we know is that the 2 copies of the same chromosome, the 2 capital letters and the 2 lowercase letters, always stick together during the first division. Other than that, their relative orientation relative to where the cell's going to divide doesn't matter. So either of these is equally possible. Both of those happen during meiosis. Half the time, the big A's and the big B's will segregate together into one daughter cell. The other half of the time, the big A's with the little b's, and therefore the little a's with the big B's, like is drawn on the bottom up here. So half of the time meiosis happens, you get the arrangement on the bottom, the other half, the arrangement on the top, by chance. And the outcome of that, this is why we wind up getting all of those random combinations of alleles that you see when you have an individual that's big A over little a, big B over big B, big D over little d, all that nonsense that I wrote on the quiz, is because the chromosomes are lining up independently. It doesn't matter if there are big A's and big B's in the same cell or not. So in the end, you wind up producing, as is shown here, from those initial cells, this is before meiosis 1, right before the first division. Those are the 2 different alignments. These are the gametes that get produced. So you either get big A's with big B's or little a's with little b's at the same time from the same meiosis, or you get big A's with little b's and little a's with big B's. So if we've got 2 genes, A and B, and each of those has 2 versions, capital and lowercase, that's 2 times 2, or 4 different possible combinations of alleles that will be produced by the gametes. And those 4 types will happen in equal frequencies. So you get 25% of each of those 4 different types. So that's independent assortment of genes into gametes. And now it's time to make use of that knowledge again. Tried it on the quiz, time to apply. Last time we were together, where we ended was crossing roller worms and GFP worms. And at the very end, we decided that we had two different genotypes of individuals. Each genotype is in one of the two boxes in the Punnett rectangle. And I asked, which one of these genotypes do we want to continue using, or what's the next thing we do if we want to make the goal genotype, being homozygous or pure breeding for the GFP mutation, the asterisks, and roller, which has to be homozygous recessive, minus over minus. So which of these, which cross, what should we do next? We've got four different individuals. We've got this parent and its genotype to use for crossing. We've got that genotype, and we've got the two genotypes of their offspring. What now? We want to make this. If we want to get asterisks and minuses into the same organism, which of these four individuals are we least likely to use? We want the roller worm parent, the GFP worm parent. Does this worm do anything for us in terms of reaching this goal? No. I mean, it's got one minus allele that it could contribute to an offspring. That's only one out of four alleles. We're looking for asterisk over asterisk, minus over minus. This worm has one of the four alleles that we're looking for. Which worm would be more useful to try to make individuals that are asterisk over asterisk, minus over minus? Well, this offspring has two. We've got one asterisk and one minus. So what are the four different things we could do at this point? There are, to my mind anyway, you might come up with more. There are four different crosses that we have to consider if we want to figure out how most efficiently to make worms that you will guarantee me every single individual would be GFP positive, it would glow green, and it would be a roller phenotype. So the first suggestion was what? Do a little incest, cross brother and sister together. That's called sibling mating. So you take the two individuals that are inside the Punnett square and you mate them together. That's one approach. That's one cross we could set up. But we discussed that that individual in the right square doesn't really have a genotype that's useful for getting to where we're going. What else would you do? Let's just brainstorm. Who else is on here? Who do you want to cross together? Pick a cross, any cross. They're all just as good for discussing. What do you think? Who's got the most asterisks and who's got the most minuses? Let's cross them together. What cross would that be? Ding, we're busy right now thinking about how to set up a cross. What cross are we going to set up?
Just top left and the bottom, one of the bottom ones.
So one of the bottom ones. So what do you want to do with, we can cross or we can self. You can self that individual. What happens if we mate this individual with itself? Cross a brother and a sister together. Will that make us any individuals that have two asterisks and two minuses? That's the goal. If you have a dihybrid and you self it, will you make that genotype? What do we need to do to find out? Let's start with that cross. Let's self this individual. What do we need to do? What technique do we use to figure out what sort of genotypes it will make when it selfs? Hey, look, we've got 25 minutes, and this is the third to last slide, so I'm willing to go slow. I think the last slide probably was what to do for next class, too. So it's this one, this is it. This is today's content. If you want to figure out what happens when you self this individual, what do we have to do? Punnett square. See, what was the genotype of that individual? I forgot already. Dihybrid, right? It was asterisk over plus, plus over minus. Is that right? Okay. So how do we set up this Punnett square? Let's do what percentage of the gametes that are produced by this cell, that's before meiosis, what are the 4 different gamete haplotypes it makes? 2 different genes. So half of these gametes are going to have what? Start on the left. We're talking about the GFP gene on the left side of each genotype and the roller gene on the right. So they're heterozygous at GFP, heterozygous at the roller gene. How many asterisks do I put in the gametes? So we can put an asterisk here. Okay, what do I put in the next? Let's say this one happened to inherit a plus. This is all GFP gene. This is the first gene, the gene on the left of the genotype. Asterisks and pluses. What do I put in the other 2? Mendel's first law, segregation of alleles into gametes, tells us that what fraction of the gametes are going to have an asterisk? 50%. Okay, so 50%, half of the gametes, we're drawing 4, have an asterisk, the other half have a plus. Mendel's first law. So for any single gene, the alleles segregate or assort independently into the gametes. Segregate into the gametes, 50-50. So now you have to tell me, where do the pluses and minuses at the roller gene go? Let me recode those as a different color so that it's a little bit easier to keep track. So where do you want me to put the pluses? I'll stick a plus there. Where does the other one go? Mendel's second law, independent assortment. Doesn't matter which allele at the GFP gene the pluses and minuses go with. So what do I put in this next one? Is that going to be a minus? Okay, put a minus there. So those are 2 complete gamete haplotypes. Asterisk plus and plus minus. Now there's no choice left. You have to give me an answer. Which symbols go in the other 2 gametes? The asterisk go with plus or minus? Minus. It has to, because we've already got an asterisk with a plus right here. So independent assortment means if at one gene there's an asterisk, it doesn't matter what's happening at the other gene. You get 50-50. Half of the asterisk carrying gametes get a plus, the other half get a minus. By the same logic, to make Mendel's first law, segregation of alleles happen at the red gene, the roller gene. Half of the gametes now have pluses, half have minuses. At the GFP gene, half of the gametes have asterisks, half have pluses. That's the top of the Punnett square. Those are the haplotypes of the gametes that that dihybrid individual produces. If you want me to be really explicit, I guess I would have to put the semicolons in. What should I write down the side? Something different or the same? Yeah, say it louder. Yeah, the same. You didn't say it louder. You got quieter. It's the same thing. When you cross an individual, when you cross an individual with itself, you're using the exact same gametes. So that makes it easy. So down the side, the 4 rows of the Punnett square, uh-oh, I made myself a small bottom row, have exactly the same haplotypes. Because I believe in efficiency, let's not fill in the whole table. Where are we going to find genotypes in this table that do what we want, that make green rolling worms? What sort of genotype are we going to look for? What combination of alleles? We absolutely have to have what to make a rolling worm? Minus over minus, right. So in which cells here are we going to get minus over minus? Only the ones where the gametes carry minuses, and only the cells where the columns and the rows that have minuses intersect. In every other cell, it'll be minus over plus or minus over minus. So it's only those 4 middle cells that are going to give us the type of genotype we want. So those 4 genotypes, in the middle of this table would be roller worms, homozygous recessive. They're going to corkscrew when we put them on a petri dish. Of those, we're just saving time. You could fill in the whole Punnett square and do this, but this might be a little bit faster. Of those 4, which ones are going to be green fluorescent? Upper left, upper right, lower left, lower right. Does it take 1 or 2 asterisks to be green? Just 1, because it's dominant. So which of those 4 squares are going to be green rolling worms? Well, this one's plus over plus. That's plus over asterisk. Like that. Fill down, fill across. Put in the semicolons just for fun. Do any of those look like the genotype we're trying to get? Pure breeding, green fluorescent roller. Which one? Bottom right. Damn straight. There's the genotype we wanted. Nice work. Now what I'd suggest you do, maybe, fill in the whole square if you want. Convince yourself that that is the only combination of those 4 gametes that are on the side and on the top of the square that produces the genotype we want. Now is the really important question. Once you get here, what fraction of the total individuals that are produced by this cross, represented by that whole square, what fraction of them are that genotype? That is, how common is this genotype? Yeah, 1/16. It's the area of the table that's occupied by that square. So it's a 4th by a 4th or a 16th of the table. 1 out of 16 squares has that genotype. It's not great, but at least we got some individuals that have the genotype we're looking for. Now what our job as geneticists is to figure out, so this was 1 cross. On the previous slide we talked about, and then I waited and waited and waited. What sort of cross would you want to set up to do this? So this is one example. You can self a dihybrid and a 16th of its offspring will be homozygous for that genotype. You go back to the previous slide. Are there any other crosses that would do better than a 16th? That's the goal of using Mendel's second law and Mendel's first law in genetics. Which way is better? If you're paying somebody in a laboratory to cross individuals with each other, if you're a farmer and you want to combine traits, and you know something about the dominance of the traits, you don't want to waste a season trying to make crosses and grow individuals that you think have the traits that you want. You want to know in advance, am I going to make 25% of the offspring are going to have the trait combination I want? Is it a 16th? Is it worse? That means drawing a lot of Punnett squares and comparing them. Does anybody have any questions about this? Sir?
Would it have been the same if we had done like the ox that was a cross, or technically sibling mating?
Okay, so what would have happened under sibling mating? Let's see. So let me write down the genotypes of those initial siblings. So the GFP was asterisk over plus, and then there was a plus over minus. That's the worm we just selfed. If we mated it to its sibling with the other genotype, plus over plus, plus over minus, do you predict that we will have any individuals at all from this cross that even have an asterisk with a minus, much less being homozygous for either? Is it possible to get one of the 16 offspring that has the asterisk from the left parent and the minus from the parent on the right? Or is that too unlikely? What's your gut feeling? Is this going to be better or worse than the cross we just set up? Can you make an individual from this cross that's homozygous for the asterisk? What would you, yeah, I don't think so. What would you have to make an individual from this cross that's homozygous for the asterisk? Has to be in, the asterisk could be in both parents. It has to be at least one in both parents. So I guarantee you this cross will not make anything that's got an asterisk over asterisk, because this parent doesn't have an asterisk to contribute to its offspring. But we can still set up the cross and see how much worse if we were really interested. So how many different gamete haplotypes will the parent on the left produce? This is the one we just worked with. How many different haplotypes? What was true on the previous Punnett square? How many columns and rows? 4. We had asterisk with plus, asterisk with minus. That's those combinations. And then we had the plus at the 2 different loci. So plus, 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 minus. That was one axis of the Punnett square. In this cross, what's going to happen down the y? How many different haplotypes does that individual make? This goes back to the quiz we started with. If you're homozygous at one locus, 100% of the gametes are going to have the plus from the first locus, GFP. So everybody's going to get a plus. So now what do we do with the plus minus at the roller locus? Alternate. Sure. So there's a plus, there's a minus, there's a plus, there's a minus. So Mendel's first law is still working. Segregation of alleles into the gametes. Half of them got a plus at the roller gene. half of them got a minus. This is where we turn the Punnett square into the Punnett rectangle, because this is exactly the same thing as that. Plus, 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 minus. So we've just duplicated information. Don't have to do all that work. So it's a 2x4 Punnett square, let's say. Just saves you from having to fill in 8 more squares if you actually want to copy across and down all of the haplotypes to make the genotypes. So it's true that we don't make any homozygous asterisk individuals. Do we at least have any individuals that have, that's the squares we have yet to fill in, genotypes, the individuals, those 8. Are any of them going to at least have 1 asterisk and 1 minus? Are they going to be on their way to becoming green fluorescent rollers? Well, all of these individuals get asterisks, and all of those individuals get minuses. Not drawing in the rest of the alleles. But a quarter of the individuals from this cross will have a combination of at least 1 minus and at least 1 asterisk. So we're partway there, but none of them are homozygous for both. So to this point, that cross we did earlier with the bigger Punnett square, selfing the dihybrid, gave us 1 out of 16 individuals that was the desired genotype. This cross, 0 out of 8. So if I asked you on an exam, which of these crosses would you rather do, which is a question I'm likely to ask on an exam, this is how you do it.
But you can always go back to the other one if you just self one of the progeny. You can just, but it just takes longer.
So exactly. So let me write out the full genotypes of these. Let's see. That's asterisk over plus, semicolon, plus over, oops, that one's minus over minus. What's this genotype on the left? Asterisk over plus, there it is, plus over minus. So we've remade more dihybrids. Then you could self one of those and get the 1 out of 16 individuals that's got the genotype we're looking for. But it's an extra cross. And we're trying to reduce the number of generations of crosses we have to use to get the genotype we want. So why wait another season to grow, or another generation of worms, to grow the genotypes you're trying to get? We could get it earlier. We're impatient. Give it to me now. Other questions about this? We'll have more opportunity to practice using Mendel's second law, Punnett squares, predicting outcomes, comparing cross designs. That's what this is all about. So we essentially did what was going to be on this slide. So that was setting up that cross that's shown in the F1 generation. So this is what to do for next class. Next class, we start a totally different subject. Linkage and recombination. So there is a reading to do. One section of one chapter, if I remember correctly. But also, for that class, please, 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 your silence means something to me. Practice working on this. Bring questions about applying Mendel's second law, and his first law, too. Here's a way to practice. This is a cross. Just draw the Punnett square. What happens when that F1 individual selfs? And fill in the Punnett square and figure out the genotype and phenotype ratio. So how many of each genotype in those 16 squares, this will be a 16 square Punnett square, how many of those squares contain the same genotype? And then convert genotypes into phenotypes. How many purple flower long pollen plants? How many purple flower round pollen plants, and so forth? So genotype ratios, phenotype ratios. Before you do that, though, I suppose we should discuss one thing because their terminology is not quite the same as the one we use. So how would we redraw the genotypes from this textbook into a format, the syntax that we use? So we'd add a semicolon between the P's and the L's, because those are two different genes, and then we'd add slashes. So when they write just capital P, capital L, that's the same thing as this notation that we've been using in class. So it's a diploid flower, each locus has two alleles, and we're talking about two different genes. So here's a dihybrid. Big P over little p, big L over little l. So draw the Punnett square for selfing that F1 individual. Phenotype ratios and genotype ratios, and be ready to discuss that at the start of next class. Then we'll move into recombination and genetic mapping. Okay? Enjoy your afternoons. Come up if you've got questions, and then of course my office hour starts now for the next hour and a half.